Question. I don't know. Okay. It would be logistically difficult, right? Well, we don't have an electron microscope. We do. It would be expensive. Viruses aren't the most stable things. Yeah, tissue culture costs a lot of money. Nobody really wants to teach it. But if you're really that interested, start demanding it, and maybe 10 years from now, long after you're gone, somebody will start a virology lab. Okay, so what's the big take-home point about viruses?
They're small.
They're small. And they say I'm allowed to teach. So viruses are small. So viral genomes are small. And so it's easy to get the entire genomic genome sequence of a virus. It's in fact these days child's play. I'll show you in a minute. So back in the olden days, there were ways to sequence DNA that really sucked. In 1971, a method came about, Sanger dideoxy sequencing. I don't think we'll talk in detail about it, which allowed for chain termination at every base, allowed for very ready sequencing. And so the first DNA ever sequenced, this was somebody's PhD thesis, by the way, was 12 bases of lambda DNA. The first genome sequenced was 5x 174, which has a grand total of 11 genes. And then stepping through time, you can see that 48 kilobases is very, very small. It's something you could sequence by hand within a couple of weeks. Automated sequencing has, of course, made that much easier as we go along through time. The largest, which is among the largest, which is 1.2 megabases, was sequenced in 2011. Human genome was completed. Thousands of genomes have been completed. They're all publicly available, except, actually, incidentally, those of you reading the book, the 1918 flu that killed 100 million people, perhaps, was not sequenced until a year or two ago. It's a very interesting story of people digging through permafrost to dig up bodies of people who died in 1918, re-isolate the virus. Somebody did that, they sequenced it, and then they published it. Well, that's what you did. Hey, I got a new sequence, let's publish it. And then it occurred to somebody that's probably a bad idea, because you could very easily make the flu again, because it's a small genome and it's easy to synthesize. So you can do it in your garage if you wanted to. And so they pulled the paper back, but it's out there. And incidentally, a couple of labs have since made the 1918 strain of the influenza in the laboratory in order to study the biology of it. But oh crap. I'm sorry, I've got the wrong file here. I was going to do something mildly interesting. To contrast that with I had a picture from our lab.
You might get
Right. That's not a very good picture.
Isolating males and answering people.
Sorry, I brought the wrong version of my PowerPoints. I was just going to point out that this would be a difficult thing to do by hand. Automated sequencing coming about has given us the ability to get genome information on viruses, not only viruses, but more complex organisms. And it's so commonplace now that even we here at Kennesaw have what's called a next-gen sequencer. We have it hasn't actually been used yet, it was just installed. We have an ion torrent personal genome machine. It's in my lab if you want to, well, it's in the room I'm in, it's somebody else's if you want to take a look. Feel free to drop by. This gives researchers at Kennesaw State, no less, the ability to do something like 40 million, round number, base pairs per day. Problem is that costs a lot of money, but the ion torrent, again, I had a picture of it, I failed to bring the right file. The ion, but I've got it. It's on my phone. I have a really short attention span. It operates on a chip, so you do mini sequencing reactions all at the same time, and the instrument can read lots of different reactions, so that you can do entire genomes on the benchtop in one lab. This is sort of a theoretical maximum operating capacity, so this stuff's really easy to get to. Within your lifetime, certainly how to get the human genome was a big deal. The field has changed so much that now you can readily get genomes at KSU, so the multiple tens of billions of dollar investment for the first genome is now laughably out of date. Except to say that's an example of using fluorescently tagged molecules in light microscopy, so you can label viral proteins either with an antibody with a fluorescent dye or with the fluorescent protein itself. And that's it for chapter 2. All right, I polled you last time and said how many people would feel comfortable drawing a peptide bond. I think I got one hand. And I don't think anybody was comfortable in drawing a nucleic acid. So I'm going to make you draw peptide bonds and nucleic acids on the exam. I want to stop and get into some atoms for a few minutes, because as we start to talk about viral structures and functions, we need to have a good feel for the underlying molecules, because they're the reason why the structures are what they are and the molecules are what they are. So there are 4 classes of biomacromolecule. They are proteins, nucleic acids, lipids, and carbohydrates. I want to talk a little bit about the structure of the first 3. Since viruses don't really do any living on their own but replicate, they replicate utilizing the information stored in nucleic acids. They have proteins that are either enzymes and do things or are structural. And then some of them have envelopes that are composed of lipids. Carbohydrates are the fuel molecules. It's the cell that provides the energy to the virus to replicate. So I'm not going to talk much about carbohydrates, but I'll tell you all you need to know. They're hydrates of carbon. But I do want to spend a few minutes here talking about the basics of biomacromolecular structure. To do that, you got to talk about the building blocks. So proteins are made out of amino acids. So, polymers. What's an amino acid? What makes an amino acid an amino acid?
When you have an amine group bonded to an alpha carbon.
It's not hard.
Carboxylic acid.
Yes, you have an amine on an acid. And so they're polymers not just of amino acids, because amino acids can be lots of different things. They are polymers of alpha amino acids. So an alpha amino acid is an amino acid that has an amine on its alpha carbon. that's the alpha carbon. This is the carbonyl carbon. It's about as far into organic nomenclature as we're going to get, so rest easy. So an R group, it's the chemistry of the R group that gives the polymer of amino acids its structure and function. But the basic structure is amine, which at physiological pH is going to be charged, and a carboxylic acid that is actually the carboxylate conjugate base form at physiological pH. And so we have a zwitterionic molecule with some R group that can be as simple as a proton in the case of glycine, or be an aromatic compound, for example, with phenylalanine. And there are 20 naturally occurring amino acids. There are other modifications that alter the structure of that amino acid. So a peptide bond is an amide bond between amino acids. So we have a dehydration reaction where we're moving water to form an amide bond, so that we have a carbonyl carbon bonded to an amide nitrogen. And that has, so the peptide bond is one big reason why structures of proteins are what they are. So peptide bond is exactly that. It's an amide bond between amino acids. We have the amide group of one amino acid and the carboxyl group of another amino acid. It has partial double bond characteristics. What's this? What does that little circle mean?
It's area where you have resins.
Right. So we have this, and the scary resin. Resonance isn't so scary. Benzene is not a pool of structures where the double bonds alternate. It's not in equilibrium. All these double bonds within benzene are equivalent. That's what the little circle denotes, is that you have bonds. They're not single bonds. They're not double bonds. They're partly double bonds. The same is true for a peptide bond, in that this the negative charge distribution is throughout the carbonyl, oxygen, carbon, and nitrogen. So this is partially negatively charged. This has some double bond characteristics of sharing an electron there. So what's the difference between double bonds and single bonds? 2 bonds. What else? They're so they're shorter. So if you look at a peptide bond, it's shorter than a single bond. It's longer than a double bond. Why? Because it's partially double bond characteristic. But the most important characteristic of a peptide bond is that it shares with double bonds. What else do we know?
It uses 4 electrons.
So what can't you do around a double bond that you can't? So right, there's no rotation about a double bond. I'm sorry, you're not Jessica, are you? Nicole. So brownie points for Nicole, which she'll be sad to learn worth nothing in real life. You can't rotate around the double bond. So proteins are linear polymers linked by peptide bonds about which one cannot rotate. So as we see a linear chain of amino acids, we can think about them as rectangles joined at the corners. So here we have a so, and since, by the way, since it's a dehydration reaction, we call amino acids in proteins, actually not amino acids anymore because they don't have the acid group, they're amino acid residues. They're what's left behind after we remove water. So here we have an amino acid residue. There's the nitrogen, alpha carbon, carbonyl carbon. So the peptide bond is, is here. So there's carbonyl group, amide nitrogen, carbonyl group, amide nitrogen. Because there's no rotation about this bond, we have a planarity between the alpha carbon, carbonyl oxygen, alpha carbon, and the amide proton. And that side chain is either down relative to the plane of the board or up. We do have rotation around the other two bonds in the amino acid, in what we call the backbone. And so a linear polymer, trying not to get too biochemistry. Chain of amino acids is going to be able to form conformations because of the rotation around the other bonds, phi and psi, which is going to position the side chains, the R groups of the amino acid, in different places. So there are 20 naturally occurring amino acids. There are theoretically 360 degrees of rotational freedom. So if I took two rectangles, and if you can imagine my hand's a rectangle, I can rotate them like this. Practically speaking, electron orbitals run into each other, so not all angles are allowed in space. But there are a very large number of possible conformations of any two sets of rectangles. If you get a protein that is several hundred amino acids long, you can very quickly get up to mind-bogglingly large possible conformations of the protein. So if we take a real sequence of amino acids, pyrosyl, glycyl, glycyl, phenylalanine, leucine would be the common name of this peptide. We see that we have our backbone atoms, our peptide groups, and then side chains that, of course, vary depending on the amino acid residue that's in there. And then they fold up in various ways to form various complex three-dimensional shapes, the most simple of which is called secondary structure. We've had all of this at some level in general biology. And that the most of the secondary structural types will even be naming, this is secondary, is the alpha helix. So it's actually a regular set of angles between those phi and psi bonds in which the side chains point out. And the rest I'll leave for Chem 3500. But we get a cylindrical sort of shape with the groups of the side chains pointing out. And so those groups can be simple proton in the case of glycine, they can be acids, they can be bases, they can be hydrophobic, they can be hydrophilic. They do chemistry. They have physicochemical characteristics. And then they do things like fold up in our tertiary structure, so into more complex three-dimensional shapes. So the overall structure of a protein, anybody have a guess as to what protein this is? It's not viral.
Is it a zinc finger at all?
It is not a zinc finger. Zinc is never purple. So this is a heme. That was a set of trap. No, wrong! It's myoglobin. So myoglobin is the protein in your muscle that binds to oxygen after your hemoglobin in your blood lets go of it. But it just happened to be the handy structure I had. I'm just showing it to you because we have secondary structures folding up into three-dimensional tertiary structures. The overall shape of a protein is called the fold. So this is folded myoglobin. And all of those alpha helices are there in order to bind a prosthetic group, so this is not protein, that binds an iron. The iron is there so it can bind oxygen. The take-home point here is we go from amino acid 1, amino acid 2, amino acid 3, we have 20 building blocks, and we come up with an incredibly complex structure that does something, in this case bind oxygen. In the case of viral coat proteins, they're going to form a capsid into which we can package nucleic acid. But because of the complexity inherent in the polypeptide, proteins are good at forming different structures and doing different chemistries. The same is not true of nucleic acids. This is why I got out of molecular biology and into biochemistry, because nucleic acids are boring. So my take-home point here is proteins do stuff, or they are stuff. Right, an enzyme catalyzes a reaction. That's covalent chemistry. A coat protein is a home for the viral genome. What's the job of nucleic acid? Information storage. And retrieval. It's not simply good enough to have the information, you've got to be able to get to it. So in the larger picture, this is DNA, this is RNA. There are, as we'll learn, viruses that store their information in RNA form. There are reverse transcription events that use DNA to retrieve it. But in the central dogma of biology, we go DNA to RNA to protein storage, retrieval, stuff. As Nick, here in the front row, pointed out, as Carlos in the front row pointed out, is there a Nick in the class? Well, there was zero chance it could be right. As Carlos pointed out, this is a lie, because nucleic acids also do stuff. There are catalytic RNAs. We'll see there are transfer RNAs do stuff right there. But in global theme here, I want to contrast structure and function. So the structure of nucleic acids is more regular, as we'll see in a moment. Amino acids, we have 20 different building blocks. We have an array of physicochemical characteristics. For the most part, nucleic acids, which are linear polymers, they do give us new ones, but I'm just too lazy. Like peptides, they are linear. By linear, I mean unbranched, so they don't. The carbohydrates will branch. DNA and RNA are linear polymers of nucleotides. So let's start at the basics. Why are they called nucleic acids? Anyone? It's a really easy question. Hey, we're isolated from the nucleus. And they're acids. Joseph's going to get the 1A. I'm going to get us. Just kidding. So here's where carbohydrates come back. We have a carbohydrate moiety. It's a pentose, so it's ribose. That has 5 carbons, 1, 2, 3, 4, 5. Carbohydrates, as you'll learn in biochemistry, have a tendency to cyclize. And so we have what was a hydroxyl oxygen of carbon number 4 forming a bond with the alpha carbon of ribose, and thus have a circular, a cyclic carbon ring. So hydrates of carbon, it's no longer, it's no longer a carbohydrate because it's not a hydrate of carbon here, but when it was ribose, before it became a nucleotide, what was my point? It was a carbohydrate. And so we have a aromatic base, a nitrogenous base, so they're nitrogen, so we have an amide bond here. It's and it's called a glycoside because it's from a carbohydrate. Carbon 1, carbon 2, 3, 4, 5. So all nucleotides have this structure. DNA and RNA, of course, vary by presence of a oxygen. So DNA deoxyribose is missing an oxygen at the 2' carbon. As I pointed out, these are primes because the non-primes are in the ring. And they're all linked by beta glycosidic linkages. So there's a structural simplicity and a regularity here. This is in the beta form, so there's an anomeric form that could be in the alpha form. Relatively speaking, the ring is like this and the base is up. The base could also be down. There's no chemical reason why it shouldn't be. Biologically, it isn't. It's up. Beta is up. Beta seems like it ought to be down, but it's not the case. Beta is up. And of course, we have 4 different bases, A, G, T, C. T replaces U, uracil replaces thymine in RNA. And then our unbranched polymer is linked by, here, 5', 3' phosphodiester bonds. So, and what makes a nucleotide is the sugar, a phosphate. We can have triphosphates, diphosphates, monophosphates. We have nucleotide triphosphates going into building. We will talk much more about this when we get to transcription and replication. But 2 of the phosphates leave a triphosphate. They make a very good leaving group and make synthesis essentially irreversible. So this is a very, DNA is a rather easy molecule to make from an enzymatic point of view, from a chemical point of view via the enzyme. But we have a linkage between the 5' oxygen, the 5' carbon via phosphodiester bond via what was its hydroxyl group to the 3' hydroxyl of the next one in the sequence. So what we have is a linear polymer with one of 4 different bases, and the order of the bases is what is the information within the DNA or RNA. And again, the difference is not only the uracil versus thymine, but the presence of an oxygen or hydroxyl at carbon 2' versus presence versus absence. And RNA, so in the central dogma, DNA is the storage molecule. It's much more stable. RNA is the retrieval molecule. It's a messenger. And when you're sending a message, the message has to both be sent and then stop being sent. When somebody's talking, if I just went, that doesn't convey any information. If I go, I don't know if that conveys any information either, but it might. The presence of an oxygen here allows for a much more reactive, a much less stable molecule because the oxygen can attack the phosphate ester bond, and it's less stable, so the molecule will fall apart faster because this hydroxyl is reactive. So DNA is, generally speaking, a better storage molecule. RNA is the better retrieval molecule. And of course, the information stored therein is in the aromatic, the nitrogenous aromatic bases that vary. You know, the mnemonic devices here, cut, pi. Sure. What's the other one? Pure is gold. No. So the purines. So clearly, not everybody knows it. So purines and pyrimidines, the 2 different kinds. I always think the larger ones have the smaller name, but that turns out most people don't like that. Purines is pure as gold. So the purines are adenine and guanine, and you cut the pi, C-U-T, pyrimidines. Anyway, I will not ask you to draw these in atomic level detail, save that for 3500. But I may ask you to deal with something like this, so that we can simplify what we think of in terms of DNA and RNA as sugar phosphate backbones that are all the same. Right? This surface looks the same to an enzyme that has to bind it, for example. And then the difference is going to be in the order of the bases. And so in double-stranded form, both RNA and DNA can form double-stranded molecules. We form a helix, a regular repeating unit of three-dimensional structure. And you've taken genetics, so I presume they go over major, minor, groove. The point I want you to think about now is, despite the fact that this molecule can be millions of bases long and have a lot of information, regardless of what that information is, the order of the bases, it looks like a circle. That's a gross simplification, but this is looking down. So take this molecule in your head and rotate here. You're looking straight down the DNA molecule. Regardless of the base pair, if it's an A, T, woo, or GC, it occupies essentially the same area. So that when we talk about information storage and retrieval, you retrieve it by going in and transcribing, for example. So that enzyme, RNA polymerase, has to be able to copy nucleic acid regardless of the sequence. It's helpful from a biological and chemical point of view that to a polymerase it's all the same thing. So I wanted to contrast the myriad of possible structures in any given polypeptide sequence that forms a protein versus the one structure that double-strand DNA is going to form. And those structures allow for the functions. What is the function? Storage of information. It helps to have a regular, you can think of it as a file format. You have an MP3 player. You're showing me myself?
It's recording.
Don't make me watch it. Come over here. Where was it? On YouTube. It doesn't help you to have a tape player if you have an MP3 file. So the DNA is a common, or nucleic acids is a common information storage format. That is not true of proteins. Okay, and then lastly we come, we're not going to talk about carbohydrates, we come to lipids. Somebody define lipid for me. What's a lipid? It's biology major. It's a fatty acid? No. It has a fatty acid. Actually it doesn't, it has a fatty acyl.
Two fatty acid chains bound to glycerol.
Also wrong. You have just described a phospholipid, which is a type of lipid, but that does not define lipid. All phospholipids are lipids, not all lipids are phospholipids. Anybody else care to try?
Has a hydrophobic part and a hydrophilic part.
So that's a description.
Four molecule?
It has a hydrophobic part.
Hydrophilic part. Can't remember. what it's
Phospholipids do. Somebody name another lipid. Cholesterol. Also has a little bit of hydrophilic. Sphingosine. Sterols. Oh, sorry, I'm not impressed anymore. Professor, I ought to be able to name a couple of phospholipids. We're going to talk about so I'm asking this because the definition of lipid is really crappy, and it's very unsatisfying. But I'm going to give it to you anyway. It's a biomolecule insoluble in water, but is soluble in nonpolar solvents. That's a crappy definition, because I can come up with things that aren't lipids that fit that definition. Well, that's not really a biomolecule. It's an organic molecule. It's anything made. But there could be a protein that's soluble in a nonpolar solvent, a nonpolar amino acid. So, crappy definition. There are a lot of different types of lipids. We're fortunate that we're working with viruses, which are small and simple and enveloped in phospholipids. So let's talk about a glycerophospholipid, which is what we generally mean when we say phospholipids. I can't blame you for saying it has a fatty acid because the word fatty acid is on the board. This is a fatty acyl chain, so glycerol is a triol, has 3 hydroxyl groups. It's also no longer glycerol because we have an ester bond between what was a fatty acid and the glycerol. So it had a carboxylic acid or carboxylate group. We form an ester bond with a hydroxyl oxygen of the glycerol, such that a phospholipid will have 2 fatty acyl chains, a glycerol backbone, a head group consisting of a phosphate linked to an alcohol. And so the difference among phospholipids, they make up cell membranes in our present interest viral envelopes. It is the difference between what this alcohol is, the length of the chains, and the degree of unsaturation. Some of those carbons in your acyl chain are double bonded. So phospholipids found in cell membranes are either phosphatidylserine, phosphatidylcholine, phosphatidylethanolamine, phosphatidylanoxidine.